This is TWIS, This Week in Science, episode number 957, recorded on Wednesday, January 24th, 2024. How is it that date already? I don't know. Filling in the gaps with science. Hey, everybody. I'm Dr. Kiki, and tonight on the show, we will fill your heads with vegetarians, HIV, and old fossils. But first,
thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. Science is a method of discovery. It is a practice that, above all else, requires high standards of ethics, honesty, and dedication to unbiased truth. When science is published, the observations of a few become the observations of the many, replicating and reproducing like a living organism. The documentation of research makes up the body of human knowledge. That is why it is with great sadness that I have to inform you of the death of science. Not that all science is dead, far from it, but that science is not immortal, that it can suffer injury, disability, and yes, even death. Currently, the scientific body has an infection of industry-crafted research papers, a pathology of for-profit publishing, a disease of career-dependent publication, presenting with symptoms of passive peer review, organized infiltration of editing boards, plagiarism, bribery, and outright fraud. The disease has become so advanced that the typical treatment methods of retractions are unable to achieve significant curative results. Damage done to science by unethical publishing standards is life-threatening, not just in terms of lost time in efforts to replicate the unreplicatable results, but in human lives lost to derailment of the scientific process and to derailment of future progress in many fields, to the living body of science itself, upon which the future of humanity relies. And most importantly, above all else, it threatens the integrity and standards expected each week by the listeners of This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science, you Kiki.
And a good science to you too, Justin. No, Blair. She's not here this week. She's got sickness and all sorts of stuff. And hopefully we'll get her back soon. But all of you are here. So welcome. Thank you for joining us, everyone out there. It's so wonderful to see you. Justin, it is so wonderful to see you this week after a week off. I was unable to keep the show going last week because of Portland's ice apocalypse. It was trees down, hundreds of trees down that nobody thought were going to fall. Like big old Douglas firs and power, internet, all sorts of things were out and people froze and it was cold and it was awful. And, you know, your whole opening about science and the death of science and the way things are going really struck me because I'm kind of been thinking since this last week, took me back to like the 1870s before power lines and before delivery dinners. And to a time when people were still searching for truth. Science was still defining itself as science. It hadn't been, systematized as or institutionalized as much as it has been now. And we find the same things striking our culture everywhere. And when things break down, it's just people left. And that's the thing that makes everything.
Yeah, and that's why you can't trust anything because it's people.
No,
Oh, wait,
what
we had a different take? Okay.
what I'm saying is that if people work together and come together as communities, that we can solve problems. And in Portland, and I know across the country, United States, I am being North America centric here. People came together to try and help people, volunteering at warming shelters, helping their neighbors to find heat, to have blankets, to do, these are little things, but it's also that in science, if people come together and work together to create the environment where scientists can ask questions, like diverse groups of people can ask questions, and there are resources available, and there are fewer gatekeepers and roadblocks, then science can flourish and foster a much better society.
And that's exactly how I felt for a long time. But I now think...
And now...
Well, I think there's something to the job of a good gatekeeper that's missed when you get rid of them. And I think that's part of the problem in scientific publishing right now, is that the gatekeepers kind of disappeared with the expansion of publications, with the open access journals, which on the first hand sounds like open access. Yeah, you don't need a subscription to see this, to access this. This is great. It opens it up to the public. But what it also turned into was a lack of gatekeeping and an easy way to charge money for a publication with a scientific name that was getting on the list of accreditations.
Okay, so we can talk about open science in this way, but or open publishing, because that's different from open science. Open science is the practice of making data available and open, and making methodologies open and available to as many people as possible. So like NASA making its data sets available for anybody to be able to access and crunch numbers. Open publishing is the practice of actually opening journals so that there's no paywall, so that, you don't have to pay a subscription fee. You can just read them. And there is the regulation now in the United States that anything that has been funded by NIH or the NSF or the US government generally, after 6 months, it has to be open to anybody to be able to read. But they still allow the publishers to have that paywall to make money for just a little bit of time. But now you're saying that the gatekeeping, now you're using gatekeeping. And I've worked with people who have been part of opening publishing and having journals that are open to begin with, where that's the way it is. There is still peer review. There is still quality assurance. There is still, yes, I see your face.
Oh, no. No, my face isn't saying anything. No words came out.
All right. For everybody who's listened to the podcast forever and has never watched the show on video, Justin tells a story with his face all the time. I know where he's going before he goes there.
I can't help it. My face is open access publishing.
It is. Yes. Oh, yeah. Okay. So, but this has been taken advantage of. And there are what we call predatory journals that take money from researchers, from people who want to publish, whatever, and they don't do peer review. They publish AI written articles. They publish fake articles, like gibberish. There are these predatory journals that people have taken time to start identifying and basically, say, this is bad. This article's bad. This journal's bad. These are the ones you can't trust them, right? And so I think that is more so the issue here. There is still peer review. We have preprint archives, and that's not peer reviewed, and that's work. The idea behind that was to get work out so that before it's published, scientists can get help from other scientists in making their work better so that they might have a better chance of getting published. You put it out there and you go, but the media got a hold of it and it became, and it's like preprints have become something that is accepted, right?
Hey, hang on.
I don't know. So I would love to hear more of what you have to say on this.
So I'm gonna start. I have this this rant that I was saving for the end of the. But I'll go ahead and do it. I'll move the bit all the way up to the top. And
okay.
Then,
no. Okay. So
because it's gonna it's gonna broach a couple of these subjects, and I got some pushback because
but what we can say,
this is how it's being framed, and I think the problem is bigger.
Okay, so now what we're doing is we're teasing everybody because we're going to talk about this later in the show and we're going to save it. So if you want to hear more about what Justin thinks about this issue, you got to keep listening because we have a whole show ahead and we have all sorts of stories to talk about. I have RNA where we don't really expect it, but we found it and what's it doing there? I also have HIV secrets, penguin poop, and some old fossils. What do you have, Justin? Aside from your rant.
I've got following a 14,000-year-old mammoth around. It's going to be kind of fun. Oh, the paleo diet that may have been mostly vegetarian. And the world's first successful embryo transfer in rhinos is making it so that the last two surviving southern white rhinos, both female, will have offspring.
Wow.
What?
What?
Is that impossible? And then, yes, I'll go deeper into why the problem with science is not limited to the idea of predatory publishing any longer.
No, I can't wait to dig into it because, the problem with science isn't just a problem with science. Science is society and culture. And so that goes way deeper. I can't wait to get into it. But let's do some stories first. So, as we jump into the show here, I have to remind you that if you are just tuning in, this is This Week in Science. If you are not yet subscribed to us, we broadcast, stream live every week, Wednesdays, 8 p.m. Pacific time, and whatever time it is where Justin is, and on Facebook, YouTube, and Twitch. We're on the social medias, mostly as @TWIS Science, and you can find our website where there are show notes and links to a lot of the stories that we talk about at TWIS.org. All right, time for the science stories. Let's talk about RNA.
Okay.
We've been talking about RNA for a very long time. RNA is not DNA. Ribonucleic acid. What is ribonucleic acid? Ribonucleic acid, some people hypothesize, predated DNA and maybe after chemical processes came to light, ended up making DNA happen and maybe RNA was responsible for a lot of the processes that led to life on our planet. But anyway, RNA, mostly when you learn about it in biology, is the messenger RNA that takes information from the DNA in the nucleus, a gene that's been translated from DNA to RNA. It takes it and it goes and it shuttles it outside of the nucleus of a cell into the cell body. So like the nucleus is in the middle, the DNA is in there, it gets translated, RNA goes, I gotta go do something, and it leaves, and it's the traveling part of information, and then it goes, oh yeah, let's make something, and it finds the ribosomes, and the messenger RNA gets turned into amino acids, base pairs, and the proteins, the enzymes, the things that make things happen within our cells and within our body. We've been finding out, though, over the last several years that there are many different kinds of RNA. There's not just messenger RNA. There is all sorts. There's all sorts. And they've been learning about these little balls of like cell stuff. They get spit out of cells that like take a little bit of the membrane with. They're called exosomes, but they get spit out. And usually they contain a lot of little pieces of RNA. And people are like, what are they doing out there? And there's some hypotheses that suggest that these little exosomes of RNA and little bits of instructions might actually influence like reproduction, that they might travel along with the sperm and influence how reproduction takes place and how genes come together and how they turn into the information that becomes an embryo. But that's not all. This new study out of Yale, published in the journal Cell this last week, has been looking at what they call RNA and cell surface RNAs on neutrophil recruitment. Have you heard of neutrophils before, Justin?
Mm-hmm.
Mm-hmm. Neutrophils are involved in the immune response in mammalian systems. And neutrophils are recruited when there's an infection, when you have a cut. Like neutrophils, they go there to fight off bacteria and save you. But part of the neutrophil action is that they need to have something on their surfaces that helps them recognize where they're going and what they're attacking and what they're doing. And they just discovered that there are little RNAs that are held on to the outside of these neutrophils. They're called glycoRNAs. So sugars, glycogen, they hold these little tiny bits of RNA onto the surface of the neutrophil. And without them, the neutrophils just pass right on by. They don't even look at the thing that they were supposed to go attack.
Interesting.
Yeah. So if they get rid of those cell surface RNAs, neutrophils don't work. They're impaired. Inflammation, like the neutrophils don't do what they're supposed to do to help with fixing the endothelium. But now they know that these neutrophils, based on this particular study, are involved in, they have what they're called P glycans or P selectins. And these P selectins help the RNAs and the neutrophils affect everything. And maybe this is a way that we can start targeting better inflammatory responses, or actually helping to control out of control inflammatory responses by understanding this little mechanism on the outside of one of the cells that's involved in inflammation in our bodies.
Yeah.
Yeah. And it's just, I just think it's so interesting, this whole idea of RNA. We used to just be like, oh yeah, it just takes information from the nucleus and that's what it does. But now it's like, it's everywhere.
Yeah, that is wild. So this is completely like they just had no idea this was like the GPS basically for the neutrophils is coming all the way from the nucleus.
Yes. And so these researchers, they really were just like, what's going on? Why are RNAs on the surface of cells? What's happening? They shouldn't be there. And so they followed it to the source. And now they realize that the RNA is really like, it's like the hound dog or the the bomb sniffing or drug sniffing dogs at the airport. They help those white blood cells, the neutrophils, actually go find where they need to go.
Yeah, so that's the question: is this instruction to the nucleus saying we need this, or is this are these sort of tags that are going along looking for a specific thing and then happening to run into it. when they
Yeah,
get
so
there.
that's one of the interesting thing. They're different from RNA that's found inside the cell body,
Mhm.
but they have been migrated. They've been taken out of the inside of the cell. So somehow there's a process that they don't understand yet, in which the RNA binds with the glycogen and becomes this component that allows it to be a first responder, and it's essential to fighting disease. More to learn. Why sugar? Why RNA? What's going on? But now we know, and this is exciting and new. Exciting and new. Tell me a story.
Well, once upon a time in Alaska, there were people. And these people lived in different places. And at around the same time, there were woolly mammoths running around
Yes.
while there were people in Alaska.
I saw Ice Age.
So, yeah, so researchers, what they basically did is they got a hold of a woolly mammoth tusk and were able to get isotopic data along with DNA from other mammoths at the site and archaeological evidence that indicates where this mammoth was traveling. Sort of, they could tell it was like living over in this area, eating this stuff, and drink from these waters over there, over a 20-year lifespan of a healthy female woolly mammoth.
Wait. So wait. They're able to identify where the mammoth went based on isotopic evidence
in the
of
tusk.
like
Yeah,
stuff
and so
that they ate. And it's like, oh, well, that would be over there, and that would be over there.
Stuff they ate and water they drank from. So
What?
This is also something that we've done in humans for a long time, where we have been able to tell, like, oh, what were the Longobeards of Italy? We can tell that the Longobeards people, who were this mysterious group of mercenaries that suddenly showed up in northern Italy, and they were there for like a long time, and had a different culture from. Had replaced sort of a culture that was there in their area, and had these traditions that were partly integrated to the Roman ways, but also had these sort of more Germanic traditions as well, and Hungarian, like Eastern European as well. And by doing the same sort of technique, they could tell, oh, this group actually was born in Germany, or this one may have been Scandinavian, or this one was from more to the east of, based on isotopic evidence within the bone that preserves how it was built, right? Okay, so they did this to this mammoth, and they also then noticed that this mammoth used to visit all of the known habitats of humans, all the hunting villages and all the places where humans gathered 14,000 years ago. And so then they realized, hey, wait a second, this woolly mammoth probably isn't visiting the humans. What's probably going on is these early humans have all decided to place their camps where the woolly mammoths go for water, where they go and graze, and where they. So
that
it kind of
seems
shows,
likely. Yeah.
It kind of more shows something about humans and their hunter-gatherer relationship with the migration of mammoths. Although there is some line in here about this being, the earliest humans into the Americas were, although 14,000 years is a long time ago, there's sites much older elsewhere in the Americas. So it depends on, like, if there's you want to add another 10,000 years to that.
Yeah.
And call this the early, like, We're still talking about there's no current modern Europeans in Europe. There's no pyramids built anywhere, and we're
Right.
still gonna have 10,000 years before any or 5,000 years before any of that's even taking place. We're fine.
But okay, so if we're talking about 14,000 years ago, this was like
Mhm.
the original idea of when humans 13,000, 14,000 years ago, that was the original evidence, right? That people
No,
used to
well,
think,
no,
humans in
no,
North America, 13. No, no, no.
no.
But then they found footprints and they found other stuff and like there's
Well, the
older
original
stuff. And
depends on where you want the original. The original was
yeah,
about 6 or 7,000 years, and
okay,
then they discovered Clovis culture. They found
right.
all these artifacts, and they finally dated them to about 13,500 years. So that was the revolutionary change. And there was that guy Jean Mars, what's his name, in Canada, who's like, I got signs of human evidence 20,000 years old in a
And
cave in the
nobody
Yukon.
listened to him.
Nobody paid any attention to him. And then, yes, now they've got footprints in New Mexico, potential cave sites in South America that are 20, maybe 30,000 years old.
So I'm thinking about the Clovis culture information and kind of like where we were a while back thinking about that. And it
Yeah.
aligns really nicely with this and also with a timeline of humans as nomadic hunter gatherers. And this isn't just a one time thing. Probably this is something that evolved over hundreds, thousands of years where cultures would migrate and follow the animals and go where the different animals went. And so, I don't know, I kind of see this not just as evidence that, like they're saying, humans probably were following mammoths, but I see it kind of as evidence that this probably was something that was well established. I
Yeah.
don't know.
Yes, and this is kind of
Just saying.
interesting
That's me.
because this is sort of before the fall of Clovis culture, right? This is when the big game was still here. Would be before the Younger Dryas event, so it shows that there was a quite a bit of human activity that far back. But yeah, so you were
Before they killed all the mammoths, right?
yeah,
No.
or they died out due to the Younger Dryas events, climate change. But the mammoth tusks, it says here, are well suited to isotopic study because they grow throughout the ancient animal's life with clearly visible layers appearing when split lengthwise. So sort of like bands in a tree, right? Tree rings. The growth bands give researchers a way to collect chronological record of a mammoth's life by studying isotopes in those layers along the tusk. So then you can kind of move slowly back seasonally through time as these layers are laid down and tell what, the chemistry of the water they were drinking from or the food that they were eating.
That's amazing.
Yeah.
I love it. Like they've been trying to do stuff like that with the DNA preserved in teeth of humans, dentin, other old fossilized stratified structures. But yeah, I love this. This is a yay.
Yeah.
Yay,
And
troubleshooting scientists. Cool. I love it.
It's cool that it does it throughout the life. Like human teeth stop, right? So whenever they're doing that, like I was mentioning the study in the Longbardis and what have you, that they're telling the story of where they grew up, right? They can tell where the individual was a child, what region they may have been a child in, but they can't tell anything beyond that because once the human teeth are developed, they stop collecting that information.
Right. Our hair works. There's other tissues, but yeah, this is great.
Kevin Reardon points out most of the evidence for when humans showed up are underwater now. Yeah, and that's
a lot.
That's
and it's going to get worse.
It's going to get worse. Well, but that's the thing because humans are so coastal. We have this huge relationship with coastal waters, and that's been true throughout human history to the point where, they've somewhere between Denmark and the northern islands of the UK, it's a little bit shallow, that a portion of, I guess, the North Sea. And for a lot of history, sort of ancient-y time history, it would have been open. It would have not been covered in seawater, right? So they have found evidence. I think there were like, I don't know, harvesting seaweed or something along the bottom, whatever they do out there, and brought up spear points of some kind, right? Like artifacts from likely Neanderthals that would have been roaming the area. So yeah, there's a lot of human history that is going to be underwater because sea levels are higher than they once were.
And this is why we need researchers who can snorkel or drive submarines or build underwater autonomous vehicles.
Yeah. Marine archaeology.
Yes. That's what we need. Moving on from mammoths, we're going to go to HIV. It's not a really clean segue at all, but it does follow the through line of scientists figuring out new ways to investigate ongoing questions and problems. Researchers just published in Nature their open access article. The HIV capsid mimics karyofurin engagement of FG nucleoporins. What does that mean? Okay, researchers know that HIV, the human immunodeficiency virus, infects non-dividing cells. It gets through the cell membrane, and then it gets into the nucleus. And it has to get into the nucleus of the cell in order to get reproduced. How does it get into the nucleus? That has been a question that researchers have been trying to figure out for a very long time. HIV is large. The capsid, which is the viral coat that encases all of the viral information, is more than 1,000 times larger than what is supposed to be able to get through pores in the nucleus, in the nuclear membrane. So like membranes, they're these, these lipid bilayers, and normally they keep stuff out, but if stuff pushes hard enough, it might be able to get in. Sometimes there are pores, and the pores are molecular constructions, components that can grab onto things inside or outside of the membrane and help them through. And when it comes to these nuclear pores, these channels that they have in the membrane, there are what are called chaperones. And usually the chaperones go, "Hey, I know you. Come on in." And they grab onto whatever protein it is and they pull it through, or they help it through the membrane. In these FG, which are otherwise known as intrinsically disordered nucleoporin domains enriched in phenylalanine glycine, which is FG, somehow dipeptides. The chaperones are called karyofurin. And what they do is they bind a cargo and they allow it to come through the FG dipeptides and into the nucleus. What they found is that HIV has evolved to mimic the karyofurin. So even though it's too big, it has its own chaperone. It's like a kid with a fake ID and showing it, and the bouncer's like, yeah, oh, of course, you fit right in, come on in. And so this is a really interesting, Blair talks a lot about mimicry in the Animal Corner and in biology, generally in larger organisms when we're talking about predator-prey interactions. But I think this is just fascinating because this is an example of mimicry at the molecular level. This is how the real battles are taking place, right? HIV is a virus and it's figured out the key. It's like, oh, I need a chaperone. I'll make my own. Oh, I got something. It looks like a chaperone. I'll get in. So not only is this very, very important for understanding how HIV and other retroviruses make their way into the nucleus of cells, it can also really help us understand how to chemically breach the nuclear membrane in case we need to get drugs into it, in case we need to use it therapeutically. This is something that we might be able to take advantage of ourselves when we are designing medications that can help people.
Yeah, that's really interesting. So then I'm also like immediately like, I wonder when this sort of, like you say, this molecular arms race took place, because it likely didn't take place in humans. Okay?
Right.
Likely this happened
No,
long
long time
before
ago.
humans were encountering HIV in any kind of pandemic type scale.
Yeah. Well, the idea is that humans were hunting bushmeat, and then that was what exposed us to primates that had, from whatever they'd been exposed to, been infected with the virus. So this is probably a very long chain of animal hosts that led to humans, decades ago. But I don't know. I mean, that's the story I remember. And if I am wrong, I hope somebody corrects me, because I don't want to share incorrect information.
Yeah, well, whether it's
The
correct
story.
information or not, that's the
old story.
At least, right? So, but then it's sort of like the molecular arms race, but it could also just all have been an accident, right? Of a host that didn't have a negative interaction with HIV in some way, that this was its method of utilizing.
Well, that's how a lot of things happen, is that there is what we call a reservoir, right? There is a species that isn't affected by the virus, but they hold it.
And then it can, and it finds all these interesting adaptions and ways to survive in this host that isn't rejecting.
Yes, and it doesn't hurt the host. It doesn't help the host necessarily, but then when the host comes close to something else that's
configured a little different.
Yeah. I have to say, just sidebar tangent, I went on a rabbit hole when I had COVID over the holidays. I started looking into cell membranes and how things are transported through pores. And I took physiology. I was a graduate student. I did grad level physiology, electrophysiology. I thought I knew how things worked. And I have to say, when you really start digging, there's a lot that doesn't actually make sense. Just going to say it right now. There are these structures, the channels, the pores, that are kind of, the gatekeepers. They have charged interiors. So if it's negatively charged, then positively charged molecules might be able to make it through. Some of them need a chaperone that guides them through, that, that'll hold them and make it safe for them to come through. But there is a whole series of different conformations and different ways that molecules that you wouldn't think should go through these pores and into cells through the memory, that somehow they do. And it's a huge, I got lost somewhere because it stopped making sense. And at a certain point, it was like the pressure pushes hard and the things go in. And I was like, oh, pressure gradients. Oh my God, that's just the end of story. But
then there's also these
the
little
cells try to do what they can. Yeah,
these little tubular things that form on the outside of cells that reach out and explore their environment and then send mRNA down the tubes to go into other cells that don't have them, or it's. Life is really weird.
So
But life
much.
finds a way. It gets really strange the more detail you collect on just
We
how a
know
cell exists.
we don't know enough yet, everybody, but I find this very interesting. So anyway, moving on from HIV and how it gets into the cells, let's talk about hunter-gatherers.
This story is just this is South America. This is down in the Andean what is it?
Plateau or the
Mountain region-y
mountains?
stuff. I don't know. Where's the Andes? That sounds like it. I only think of the mountains. But this is early humans in the Andes spanning 9,000 to 6,500 years ago.
Okay.
And by doing analysis of ancient human remains, they were able to find, again, isotopic composition of human bones to show the plants, the foods, and meats, whatever, that made up the majority of the individual diets. They found 80% of the diet was plant matter, with just 20% being from meat sources, suggesting that these hunter-gatherers were likely doing a lot more gathering than hunting. It's also interesting is that they found that these early Andean people were eating a lot of potatoes, which were developed there by farming.
They were poisonous.
No, they weren't poisonous. No.
Potatoes were poisonous. I mean, that.
Wait, what?
Yes, historically, potatoes were poisonous. It's thanks to humans and their cultivation of potatoes that potatoes have become less poisonous.
So I didn't know that. I knew something like you're not supposed to eat the leaves of potato, and people who tried that got sick. Like when they brought potatoes back to Europe, people were eating the wrong part of they were eating some leafy shoots or something from the plant and got sick when it turned out, it's not the part you eat.
Don't do that.
Just
don't eat
wrong.
The green stuff.
But yeah, so it kind of suggests though that maybe some farming and cultivation was taking place even earlier than the earlier creation of these things in the Americas. But anyway, yeah, the idea of hunter-gatherers is going around hunting game all the time.
Maybe not so much.
Yeah, maybe not so much. Maybe it was. Maybe, farming wasn't actually that big of a leap for humans. Because you can kind of picture, all right, you're doing a little bit of the nomadic thing, right? Well, we're going to go, but you're chasing, not game, you're chasing plants. The potatoes grow in this season over here, so you go over there. And yeah, you might kill a couple of animals that are there to eat that happen to be there also. And you go, winter's coming, we got to go over to this place where we know this other tuber underground is going to be. We can eat that because it's still these berries that grow in the winter, whatever it is. And then what do humans do? Like every animal does. They tend to poop. And they have trash middens. They have places where they put their garbage. And they tend to do these things in the same areas. And
some of those things that they poop out are seeds that have been digested. And then they germinate.
And they grow. And then somebody notices, hey, did you notice that every time we leave this camp and we go to the other camp, we throw out our old potatoes that we took along with us because they went bad. Then we get a potato patch in that spot next year. And then it kind of just evolves from there. People making observations. But it sounds like, I don't know when we think this revolution of farming took place elsewhere in the world, but it sounds like it may have already been in place in the Americas somewhere in the neighborhood of 9,000 years ago.
Yeah, I mean, from what I've heard, I think the oldest agricultural evidence is maybe 10,000. So it's right about the same time-ish. I mean, give or take. Fascinating. People, we've been doing stuff a long time. You gotta pay attention to it.
Also, your paleo diet is mostly vegetables.
So
Congratulations on eating the wrong thing again in a fad diet.
I think you reported on something like this a long time ago where it was like more grains or something there was some other evidence. Like
in Europe, they were finding
yeah.
that the people ate quite a bit of grass and grain, and meat was also a supplemental.
Mm-hmm.
But
so this is like more evidence, similarly, but on another continent.
If we actually had the omnipotent light of all-knowing science at our fingertips, I think you could basically take every book on nutrition off the shelves. Like, it can all come down.
I don't know about all of them, but
anything that has to do with diet and nutrition, that's not a negative, right? If we put the warning labels on the ultra-processed food for the things that we know, the diseases that we know are caused by these ultra-processed foods, we could do that labeling. But all of the health benefit stuff for diet, a lot of that can come off the labels. You don't know it's not that food is healthy, it's just food. Food is just food, but it can be unhealthy food. And I think that's the difference.
Yes. No, I think you're exactly right. The psychological valuation of certain foods over others is actually we're doing ourselves a disservice, but it is good to know the kinds of food that are available to us and their nutritional value, but not give them value moral judgment. Whatever, this is food. Eat what you want. My final story for this little first part of the show, which took an hour, is that researchers have determined a molecular switch that determines whether or not osteoblasts, which are bone-forming cells, become bone-forming cells, or whether they turn into adipocytes, fat-storing cells. Researchers there at New York University, they looked at skeletal stem and progenitor cells. These are cells in the bone marrow, and they go on to help with bone development and repair and all. And they tend to go into more fat storing and less bone forming as you get older. Mm-hmm. And so something happens as you age that creates an environment that tells these stem cells to be like, you got bones. That's okay. You need fat. Have some more fat. And when that happens, you become more likely to have a fracture. This is probably part of osteoporosis and other issues as people age. They looked at the molecular mechanisms in mice, and they were able to look at the gene expression, single cell RNA sequencing, in skeletal tissue. And they found that there are specific signaling molecules that are involved in this switch. And there's a group of them. It's what they're called notch signaling genes. And so as you get older and you have bone degeneration, there's more notch signaling gene activity. And so they think this is what's active. And they were able to switch this around in the mice and change the notch activity. And what they say is that they basically showed that they were able to change the phenotype, which is how things worked in the mouse, not the genotype, but how everything came out, in such a way that they changed the mineralization, and they were able to create more mineral for stronger bones in the mice, that they were able to downregulate the notch signaling. So if they got rid of it as the mice aged, then the mice had stronger, more mineral rich bones. And so this could be a target for helping people have stronger, healthier bones as they age. That said, this is not a replacement for doing the things and eating the foods that keep your bones healthy in the first place and make sure that you're aging healthily. But if you do have a condition that is leading to bone mineral degeneration, who knows? It could be on probably available for your kids, but right now available for mice. They're fixing mouse bones.
Yeah, mice are going to get all the good treatment going forward.
They always do.
Well, this is good news, because of all of the goals that we set for aging, usually we start with, I want my appearance to be good. I want my mind to be healthy. And so here you are. You've directed scientific medical progress so that at age 150, your skin looks great. Your mind functions perfectly well.
And then your hip
bones
breaks.
are brittle as a sugar cookie. Like, oh no, we should have started with the foundation, the skeletal structure of the human before we worried about the other things. So this is good. This is another step into humans living forever.
Yeah, and notch signaling genes are involved in a lot of different things within the body. So this isn't just to be taken lightly, and you can't just be like, get rid of notch. That isn't necessarily what you want to do. But if it's approached in the right way, maybe it's a therapeutic target, or I don't know. You were talking about diet earlier, and there has been evidence also that weightlifting, or lifting stuff that isn't just your body weight, can help strengthen your bones because it's giving your bones a struggle. People who are able to walk longer distances are less likely to have brittle bones. All this evidence is to say, there's something involved in how metabolism works and what ends up creating the switch and leading to upregulation of notch genes versus youth blood.
Yeah, there's a whole the bones are just like such an amazing organ. But yeah, the more you use it, the more sort of wear and pressure, I guess, you put on your bones, the more they build, the stronger they can become. And they're also, this is, you talk about, you gotta eat right. Well, you know where most of your body's calcium comes from? Your bones. Your body uses your bones calcium.
Unless you eat lots of good food with sunlight and vitamin D and all the things, and you have a balanced diet, and then you're healthy.
Well, it's kind of a point that it's like you can eat if you don't have calcium in your diet, I suppose I should say, your body will still need it and use it from your bones. It will take it. It will take it away from your bones and use it elsewhere. I think it uses the liver might need it or something. Or it
calcium is important for muscle cell function, for nerve cell function, for liver cell function, for basically every cellular function in the body. There is a calcium gradient involved, and calcium is also involved in apoptosis, cell death. Calcium is essential. We need it.
So drink your milk and eat your cheese, folks. That's the nutritional.
Oh, I want to eat more cheese.
But then, if you're having nutrition. Okay, so anyway, but this notch thing is, it's always
nacho cheese.
It's always tricky when you find a mechanism Because then you're like, oh, what happens if I make too much nacho cheese? Is that going to be bad for the bones? What if I don't make enough? What if I've turned it off too much? And what if I turn it up too high? It's one of those things where you can, like any
You can overdo
lot of
it.
drugs, it's about getting it to balance in a good way. So now I got to study what is this notch ratio in all the healthy young people, and what does it look like in people who have bone problems, and is there a way that we can regulate to a degree where we're turning one to look more like the other if it has the benefit? And yeah.
I think you have just determined somebody's research trajectory. That's great.
Yeah, I think it would be a good one.
No,
And you know, it's a good source of research projects. Clearing your mind for a quick break.
This is This Week in Science. Thank you so much for joining us. This is a very quick break, as Justin has just said, before he tried to break his desk now that he's off of the video. Thank you for joining us for another episode of our science discussion fun. We like to have a good time here. We like to talk about science. We like to enjoy digging into things, questioning things, maybe learning a little bit of something new. And if you're enjoying this as well, and you know somebody who likes learning and digging in and trying to figure out things around the world, maybe share the show with them. Let them know that TWIS is a show that they should subscribe to. Like, help them out when you're with them hanging out and they're looking at their phone and ignoring you and, surfing the internet looking for scientific information. Just be like, I know what you need. And then, subscribe to TWIS on their phone for them. I mean, that could work. Additionally, we do definitely love your support. If you are able to head over to twis.org, you can click on the Zazzle link, where we have a link that goes to our store at Zazzle, where we have Blair's Animal Corners, TWIS 2024 calendar. It's available right now if you need a calendar for the year. And there are lots of other great products as well that help to support the show. And our Patreon link is also at twis.org, where you can click the Patreon link, get over to Patreon and join our Patreon community and be a supporter at any level that you're able to. $10 and more a month. We will say thank you by name at the end of the show. And there are lots of other fun things that get sent to you from Patreon, depending on the level of your support. But I just, you being here and listening and being a part of this whole group, you let us do what we do. So thank you very much for your support. All right, coming on back with a little bit more twist. Let's keep this show going. Dun, dun, dun. Justin, you want to tell some stories?
Well, what have I still got left? Oh, I said the southern white rhino was down to 2. It's the northern white rhino, apparently. Okay. I had the story completely wrong.
Blair would have corrected you if she were here.
Yes.
Yeah.
So it's the northern white rhino that's down to 2. And this outfit called BioRescue has succeeded in achieving the world's first pregnancy of a rhinoceros after an embryo transfer. Their new technique and method used a rhino embryo from a collected egg cell and sperm that was transferred into a southern white rhino surrogate mother at a conservancy in Kenya. Now, what's wild about this is that they don't have to do that sort of crossbreeding scenario where they're using a southern rhino to make more northern rhinos, because the northern rhino is down to the 2 females, a mother and daughter. However, they have sperm. Somebody was smart enough to collect rhino sperm before
they saved it.
Mm-hmm.
Okay, I was wondering if this was some kind of gene transfer or like I don't know what, but okay.
They
stored sperm.
They apparently have living cells from 12 different northern white rhino individuals stored in liquid nitrogen. So they're now this pregnancy was induced. What was it? September of 2023, and they are confirming that they are 7 days into the pregnancy, and I believe they've had 13 other tests, embryo transfers that they have performed. So there's, they have some decent now evidence that they can take this technique that they've been developing and apply it to the northern white rhino. So this is really a wildly international, but they've got currently they have the stored liquid nitrogen is at a minus 196 degrees Celsius is stored in Berlin and another and in Italy. So there's a couple of locations where they have the banks. This is research that's taking place in Kenya to do the proof of concept. So it's really like a lot of groups working together here at BioRescue, researchers who've dedicated themselves to conserving a species, which is really amazing. And this is where I would always like to take a moment for anybody who has a preconception of what a rhino is. They are not fearsome beasts as they are often portrayed or thought of. They actually, in their day-to-day lives, act like really big puppies. They spend most of their time playing with each other. And when they sleep all snuggled up next to each other. And they're just the sweetest creature.
Rhinos are like little puppy dogs.
They really are.
Yeah.
They frolic in the wild. They play. It kind of helps to be a big rhino who can get kind of fierce if they need to. That they probably don't have a whole lot of predators.
Can I show a picture of the embryo? Would that be appropriate? That people might like to see that. It's with the materials from the press release, and they have the southern white rhino fetus from the successful embryo transfer, September 24th, 2024. Wait, what? 2024? What year is it?
It's 23. That's a miss.
You're like, wait a minute. Is this future time travel?
Yeah. Yeah, the caption there has got somebody's been writing the current date, and remember, I gotta write the current date. Oh, it's not 2023. The text in the article clarifies.
Well, the little baby rhino looks like it's a little baby. Pretty little baby. I wonder how much this kind of technology is going to work in the future, whether sperm banks and storage will allow more species that are becoming endangered to have a chance of coming back. I mean, population bottleneck of 2 females and then stored sperm. I guess it's a question of how many males do they have sperm. Yeah, it doesn't. So
it doesn't, but I don't know if they're. But I don't know if they're related. I don't know how related they are of those 12. And then if they're even, they might even be related to these females. So it's depending on what that makeup is going to determine some genetic health going forward. But hopefully they can move forward and do this responsible way. Adea Wilson is asking, do rhinos learn like puppies? Well, I haven't actually spent that much time in.
Talk to Blair.
Yeah, we got to talk to Blair about rhino intelligence and cognition stuff. However, I will tell you that they do they are potty trainable. Mm-hmm. Rhinos, as a group, will all go to the same location to poop, and they won't poop where they sleep, and they won't poop where they eat, and they will take a little walk. Sometimes as a group. One sees, oh, you're going, I'll go with you. And they'll go, and then they'll
it's
turn
like
around.
Girls going to the bathroom at a club.
Yeah, and they'll go into the same spot as a group, they've sort of, I don't know how it's decided, but they all know this is where we're taking care of our business, and not over there, not over here, not over there, not in our play area. So they are, at least to that degree, trainable within the group. I don't know if we can train a rhino. I don't know if anybody's tried.
I'm sure.
I think somebody's probably tried. But maybe
zookeepers
would know.
Tangent, though. You want to do a rant, but we were talking about rhino poop. So I want to move on to penguin poop and then my last little story, and then we can rant. Does that sound okay?
Yeah.
Yeah. So we've talked before about watching animals from space and the possibility of different satellites to allow us to visualize colonies of animals, and specifically penguins in Antarctica. And a new study that is just out in Antarctic Science, which is a journal, has shown that by using the European Space Agency's Sentinel-2 satellite that has a spatial resolution of 10 meters per pixel, a little blurry, but it's fine. The researchers were able to identify more breeding sites of emperor penguins based on their poop on the ice. So the researchers were able to identify the colonies. They looked at a whole bunch of different spots around Antarctica using this satellite over the years. And several years back, I think, Blair, one of us reported on this news that these researchers were doing this, but it had less fine resolution. Back when they first started trying to do this, they were able to get a smaller number of breeding aggregations and colonies. But at this point, they were able to identify at least 4 more emperor colonies. And it was identified not just the penguins, but because of the darkness of the snow, because of where the penguins left their guano behind.
Yeah. That's clever.
Yeah, but this is also very exciting because this species of penguins and other penguins are becoming more and more endangered, especially as climate change is reducing the amount of ice that's available at the on the coasts of Antarctica and other southern continents. And there's a lot of habitat that's disappearing that many species of animals, in this case, we're talking specifically about penguins, but that are it's just disappearing. And so the way that these animals use ice and live on it in order for their breeding to work successfully, that way of life might be going away. And so it's good for us to know where there are and how many breeding populations there are. But with this, it brings the number of breeding populations up in a group of animals that is on the verge of getting on the endangered species list. So this is good news. Yay, looking at poop from space. Woohoo!
And penguins are really clever. They found a continent that didn't have anyone else on it.
We're going to use this. Nobody's here except,
if we just, it's going to be tough. It's going to be tough going, but there won't be anyone else here. Perfect.
Yeah. And additionally, I must specify that they also used for these 4 breeding sites the Sentinel-2 and Maxar Worldview 2 imagery to identify these things. Okay, moving on from penguins, let's talk about some old fossils. There are lots of questions about when multicellular life emerged on our planet, and we've got a lot of dates that go back. Single-celled life. There's possibly like 3.9 billion years ago, like billion. Yeah, there's some rock fossil evidence that is questionable. People are still debating it. Possibly as long ago as 3.9 billion years ago, which would have been like 500 million years after the Earth formed, which is
but there's this
wackadoodle.
big gap too, because we have that pre-Cambrian explosion thing where we got all these life forms, but the reason we have this huge gap before we see all this life is because we imagine that they were soft-bodied life forms, and they just don't lend themselves to fossilization very well.
Except that many of them have metabolic processes that involve things like calcium that we were talking about earlier, or other elements that mineralize over time. And if the mineralization of their metabolic processes is captured in that moment in time against a rock, or
just the mud
in the
bottom of
mud,
the ocean, maybe.
then those minerals can potentially be a hallmark, a sign that something living was there. And because we understand certain chemical processes, there are certain things that work in organic chemistry, certain things that don't. We know certain actual processes that go from one form of energy to another, one use of calcium or hydrogen or whatever it is, and change it to another formation, and that can be left over. We're using this evidence also to look for life on Mars and other planets. So in the lab, researchers have shown that yeast can be multicellular. And so that was kind of interesting because, they're like, yeast, we think of it as like a single-celled organism, but, it can be multicellular. And so these researchers published in Science Advances their work looking at a bunch of rocks from a formation in North China. It includes layers that are 1.6 billion years old, but there's been some dating that has taken those rocks back further. They have taken fossils out of the rocks near the same area. They dissolved them and found microscopic fossils. And in the microscopic fossils, they seemed to see cylindrical cells that were attached to each other, kind of like they had cell walls, like plants. And they think they see spores within the cell walls. So they think there were specialized ways of reproducing. So what they are saying is that they have discovered evidence that there were organisms that were multicellular about 600 million years before we think multicellular life existed based on this evidence.
Before we have evidence of.
Yeah.
Because I think we think that it's much older than we have the evidence of. It's just a matter of, again, soft-bodied creatures not lending themselves to fossilization, not having a shell, not giving a good imprint in a rock or what have you.
Yeah, so this, still debatable. People are questioning whether or not this is what the researchers think that they're seeing, but the researchers are themselves convinced that their evidence reveal these morphological details that are obviously multicellular and that can be interpreted as eukaryotic, and that eukaryotic, multicellular humans, we're eukaryotes, that our type of life and probably photosynthesis even may have started earlier than we think. A lot earlier. Yeah. I don't know. This kind of stuff is always so interesting to me because, like I said, we're basing our assumptions on chemistry that we know, on current morphology that we know, and we're looking at these things that have been dissolved out of rocks and going, this is a thing, this. But I think it's
the Earth captures these things, and if it's somewhere in the Earth that hasn't, so much of the crust is in constant upheaval and in a solution where it's dissolved or it's being pushed down or up into the elements and blown away and turned back into dust. Like, when you find an area that has been undisturbed for billions of years,
yep,
that nothing has changed. You can find evidence like this. So there's not a whole lot of places on the planet where we can do this. So this is an important site for sure.
Yeah, it's a very important site. And I think it's something that people will be looking at a lot moving forward and continuing to, question these results and really determine what they've found and what they're seeing here. I think always in science, it's based on replication and being able to come to a consensus over time with enough evidence. But this suggests that like these structures are similar to green algae, but they're so much older. And what were they doing there? What happened? What's going on? Old rocks. So, scientists looking at the world, questioning things, having all sorts of ideas. They want to publish. They get peer reviewed. Sometimes people go, and then there's a debate in the journals and they fight each other in the journals. And sometimes they fight each other on social media. And Justin.
Which brings us to a new segment on the show that feels like an old segment, but is a new segment, all the same, called Justin Rants About a Problem, followed by more hope-filled words from Kiki.
I love that. I love that addition at the end of it. I really do. I hope I'm in a mood for the hope-filled words at the end of it. Okay, go, rant.
Okay, science has a problem. Mostly due to publishing practices where publishing companies with profit motives have very few guardrails when it comes to what they publish. They have been, for years now, many publications looking the other way when it comes to fraud and plagiarism even. They look the other way because they make more money if there are more papers submitted. We were talking earlier about the new platforms over the last 15, maybe 20 years now of online publishing, or the many, 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 many journals that have sort of sprouted up out of nowhere. The increase in the amount of papers that are published, it's like 10,000 a day or more now.
It's so
and
many.
Each of these, it used to be that there was a subscription paid to the prominent journals by all the universities. They all paid a ridiculous amount of money for a thin group of papers that were published, and that's how the journals survived, from the subscriptions. And now the new wave is generating the income from the researchers submitting their papers to journals. 2,000, 3,000, 4,000 a pop, 10,000 papers a day or whatever it is now. It's just ridiculous amounts of money are flowing in there. So it's as bad because science has another problem, which is paper mills. So
right, yeah. So I mentioned predatory journals earlier, but yeah, paper mills.
So paper mills are before you get to the predatory journals, right? Because paper mills are companies that create basically fake research as a business.
Yep.
They create these papers and then they sell authorship of these fake papers to anyone who wants to be on a research paper. This is increasingly a popular choice amongst people in the medical industry, either grad students or people who are currently working and don't have time to do research because they're too busy working, but want to show that they are doing research. There's job opportunities or raises,
and it's not just the wanting to. If you're an MD, PhD, there's a requirement for the PhD part of it for you have to publish a paper. So there are like certain requirements, and
you got to
part of the
spend
system.
money to make money. Yeah. And so by paying to have your name on a research paper you have nothing to do with, along with other people you have never met that you're not collaborating with, for research that wasn't actually conducted but was published in a real journal, getting that paper cited by other papers also helps, but that costs extra. For that, you will need the citation mill upgrade package to your paper mill package.
Don't you just go on like Chat GPT for that? That makes your citations up.
Oh no, not no. So, and there's also self citation of journals that's increasingly so. So you've published. Now maybe you've published a real paper. Your real paper has been submitted, and there you get a suggestion from the editor. Hey, you know what? This reminds me of 10 other papers that we've published. You should cite them. Oh, okay. Well, I'll add those citations then. Thank you for that, editor, for giving me that helpful tip. And now that publications, other published studies have more citations than they did before, and this is increasingly, you will see a normal scientific journal will have in the range of 2 to 3% self-citation, where it's citing other studies that they published before. A lot of times, if you delve into that, it's also because they're very specifically working on, I think a lot of times people will be citing their own previous research because this is a continuation of that research and the things that led them to start that research. And if those are all published in the same journal, that's where you end up with this 2 to 3%. And the average for these new generation of open access journals is more in the 12% range, with some being as high as 20%. Okay. So
depends on the journal, right? Yeah, it's a problem.
Yeah,
it depends on the journal, right?
It depends on the journal, right? Okay, so this is a big thing. That depends on others. Some are credible, some are less so. The peer review process in every major journal even can be considered dubious at best, and the retraction rate for bad papers is slow. It lacks a way to generate money, so most publishers don't even bother with their retractions. They don't even bother with internal investigations. The vetting process for papers is now really low. Because there's like so many thousands of papers that are being pushed through, they have guest editors. These are people who may not actually even be in the specialty of the paper that they're reviewing, coming in and sort of doing the initial review of it. They have guest peer reviewers who, again, may or may not be tied to the journal itself. They may be editors who are paid elsewhere by the paper. So anyway, there's this thing called impact factor of journals. It's curated by Clarivate Analytics Web of Science Group. They use citations, number of publications to create impact factors for journals. That the citations is part of it. That's why there's a lot of self-citation going on and pressure to cite within the same journal that's publishing your work. So, but it's a form of credibility rating that they have put out. In 2023, Web of Science removed the impact factor of 82 journals, including one of the world's largest, that had a great impact factor and a wide distribution, a large number of distribution by meaning a large number of journals of papers being published within it. So 82 journals, these aren't individual research papers, but hundreds of thousands of papers published under the umbrella of journals that are no longer considered credible. Great first step, right? Web of Science has also been doing things like using AI. They've been developing AI because there's such a volume of these papers that they can't humanly read them all, humanly investigate them all. So they're now using AI to start vetting what's being published by these publishers. Many of the 82 that were delisted were under the Hindawi publishing brand.
Yeah. Yep.
And they got delisted right after they were purchased by Wiley Publishing for a ridiculous amount of something near $300 million they paid for this publishing company. Wiley began an internal investigation after being delisted, having their new purchase told, it's not worth anything.
Yeah.
They became so overwhelmed with the retraction-orama puke fest of regurgitated paper mill pulp that they had bought that they have, by the end of last year, this has all happened last year. They bought it I think they purchased it last year, got delisted last year, and they have now announced that they're retiring the brand that they bought entirely. Once they move a few hundred publications over to Wiley, which means exactly what when you're moving the publication around. But maybe Wiley's going to have a much higher standard going forward for what actually makes it into those publications. Meanwhile, the current edition of AAAS Science Journal has several stories about science fraud, a whistleblower report on a Chinese company bribing editors of publications. This is not just now, hey, you come in, here's a paper, we can put your name on it, and then, hey, here's a publication we can try to get into for it. Now we just want to bribe the editor of the publication in the first place so that they are assured to put you in, so that the whole network is complete. So this is framed, though, as a problem for, like you were saying, a lot of the junk journals, predatory journals, less credible journals. But, I've been reporting previously on junk Chinese science publishing at all of the major publishers. Yeah. All of the major publishers. As if there's a different standard for the glut of these state-sponsored papers coming from China that they would just cost too much to reject. And I'm looking at you, Nature. I'm looking at you, JAMA.
So Nature is Elsevier. So it's a different, the Nature journals are under the Elsevier umbrella corporation. And so I think, that is the corporation that I've heard people talk about a lot, especially with respect to getting rid of paywalls and making papers available to people. Coming from having been in the academic world, met and known people who have who have edited and actually run journals and worked for some of these companies. It's a really interesting ecosystem that we have created because we allowed there to be publishers, like book publishers, that need to make a profit, and they're public companies that have shares that then need to have quarterly returns. And in that, they have grown, like Nature, that journal is very well respected, high impact factor, one of the most respected. It's like Science, Nature, Cell. These are some of the top ones out there. But at the same time, not all the papers are available to everybody. And because of that, as more and more scientists fight for open information so that people can have access to information and knowledge, the publishers are, they're holding on to what they've got because they have to make their profits. And, but who's going to pay the editors? And at this point in time, when you have a peer review or a person who reviews a paper, you don't do that for money. You do that for free. So you have an editor who's probably paid by the journal, who's paid by the umbrella publisher. And that editor is looking through and goes, okay, maybe this paper, now I'm going to send them out for peer review, sends them out, basically asks scientists who have a billion other things to do to read this paper and say what they think. How is it? Is it good? Is it bad? Whatever. And they do that for free.
I don't. That may be what's going on at Cell. That may be what's going on
at Science, at Nature, at all the really good old school journals,
but not the new ones. The new ones.
But scientists have been upset about this because
they have too many.
Yeah, and scientists have been upset because they're like, okay, well, I mean, this is part of my job as a scientist to be part of the community and help the research move forward and feed back into what's going on, but they're not paid well. They have to get grants, and then they're not paid to do these reviews, and that's extra time that the university doesn't pay them for. So I imagine that there are probably a lot of researchers who are like, oh, okay, paper mill? What? Oh, it's a paper? I'll read it. And they review it, and it's fine. People need to make a living somehow these days. End stage capitalism. It's awesome.
Well, then you got JAMA, who's been sort of a pioneer of this open sourcing, right? Get the information to the people should be able to read this scientific information. They're also one of the biggest growers during this open source revolution.
Yep.
I recently asked
they have
JAMA,
a lot of you go to JAMA, they have advertisements.
Sure.
Like you go to their papers, there's advertisements on the pages. It's like I'm like old school bloggers making ad money. They're like, what? Okay.
And they're charging thousands of dollars, of course, for submissions to there, because they don't have a subscription pay side that's paying for everything.
Yep.
And so publishing it is really putting it on a website and being done with it at this point, right? But I recently asked JAMA if I could read the peer reviews on a peer-reviewed study that I had some serious concerns about, and was told that the information was confidential. Not that it was behind a paywall and I had to join some other thing, but that the information was confidential. That was used to advise on the paper and not something that was for publication. So some journals will have you can look up the peer review. You can see what the other, 2 or 3 researchers who read the paper and said, oh, I would be careful of how you used framed the data here, or did you look at this? The way you have presented your infographic could have a little refinement.
I remember my first peer review, and somebody, what other your statistics should not be this. Yeah, you're using this statistics, and you should be using these statistics, and blah blah blah. And I was like, oh, okay, I gotta go back and do it again. All right, that's what you do.
So the one I was looking at was a paper that was connected to Odyssey. Odyssey is this group of researchers and data professionals who have gotten together to use big data sets, and they also have an undercurrent of funding from the pharmaceutical industry. And this paper had some just findings and conclusions that their own data even seemed to be contradicting. So I just wanted to see what the peer review looked like on that paper, because gosh, I had some concerns that this was industry driven and that for some reason it was, the data just didn't make sense. But I
think I remember your report. You talked about this, like before you left and went, on your little sabbatical, getting a new job for a while. Like you actually talked. I remember you talking about this paper. So you actually did dig in more.
Yeah, we reached out to the folks over there and just tried to get, hey, can I see the peer review of this? That's all I wanted to do was read it, and was told it was confidential. And so for the open access, open network JAMA journal to have confidential peer reviews makes me question whether there is one. Honestly, I have now doubts about the integrity of JAMA as a whole. So another story that was in Science here recently was about, not in the journal, in the journal Science, or on the website for AAAS. It's about a dozen papers by the head of Dana-Farber Cancer Institute, the head of, and 3 of the senior researchers needing to retract 57 papers published between 1997 and 2017. This all came to light not because of any peer review, not because of an internal look within the journals, but because a blogger noticed recycled data was being used in a number of places in their studies, which could be accidents 57 times, or could be data manipulation 57 times.
57 times?
So my point is that if I should limit myself to one here, is that the scientific publishing industry standards are too low. So low at this point that I do not trust you. And I was your biggest fan. But there was like, this is one, oh gosh, where is this one going to have been published? This was published in JAMA. Okay. Where I remember hearing an interview about this because I had to go and keep looking at this. This was a trial of a Chinese medicine compound.
Oh, yeah,
This
you
is
were,
the-
yeah.
This is the
You're
one
like,
that
how
had
did this
like,
go through? Yeah.
when you looked into it, it had ground up insects and all these different and they combined it with a Western drug and said, hey, it has this positive impact.
It's like, what
And,
actually is that impact? What? Yeah.
But what is even in this compound of like ground centipede, this kind of a spider, this sort of a flower, that kind of an herb? It's
And
holistic
Justin
medicine.
is just like making up a whole bunch of ingredients right now. He doesn't remember.
No,
all the
I'm not.
ingredients.
No, wait, no. Do you want me to read it? Hang on.
Do you have the ingredients? Like,
Yeah.
is it
yeah.
really that? No centipedes and spiders. Come on.
Let's see. Did I? Oh, maybe I did. Okay, here we go. So they don't list in this JAMA article what the ingredients are of the compound,
Mm-hmm.
but I found a previous publication by the group detailing the proposal for the study to look into the compound, which did list the ingredients. And they were Chinese scorpions, leeches, Chinese cockroaches, Chinese red-headed centipedes, the sloughed outer shell of cicadas, red peony root, sour juju seed, agar wood, white sandalwood, and synthetic borneol, a substance derived from camphor known to be highly toxic. Now, these are all combined and ground up into this compound that I can't pronounce quite properly, but it's like Tong Xinlu, something like this. It's a Chinese traditional medical
Yeah, okay.
thing, right? And the compound was used with some level of ingredient from all of these things in conjunction with a Western medication to treat a thing and showed a positive input. Now, this was so amazing to me that, first of all, they would not list in their publication what was involved, what those ingredients were. Just said a list of ingredients, but that this could be considered science at that point. That this could be considered research. And I remember I delved so far into this that I got into an interview with one of the publishers who was talking about how they well, we had questions about whether or not we should publish that study, but
we did it anyway.
Were seemed so important and significant that we thought we had. No, you're paid. We can tell when you're paid. We can tell when you're paid when you're showing ground up centipedes of holistic medicine, a state-sponsored campaign in China currently having been announced as a goal by the Chinese legislative branch, or whatever you want to call it, is to introduce holistic medicines into Western medicines as add-ons that improve. And now we're seeing all these research papers that are trying to find that, and you're publishing them in your supposedly high-impact, credible journal. I have lost all respect for you, JAMA. You are delisted by me. If the Web of Science hasn't caught you yet, they will. And I'm really curious.
So here, number one, the addition of the Western medicine compound, it sounds a lot like the homeopathic medicines that are sitting on a lot of shelves that actually have things like epinephrine or, actual poisons and toxins that can hurt your babies in them, but nobody talks about them, right? Oh, that's never mind. That's the active component, but it's the homeopathy that's doing what it is for you, right? So that's number one. That's what it kind of rings as familiar as and similar to. And then the other side of it is I do not think it's wrong for Eastern medicine and traditions to try to incorporate themselves into Western medicine. Why are we separate and what are we doing? But evidence needs to be the basis and it has to be done right. And if what you're doing is putting a mishmash of oatmeal from the kitchen sink in there, plus, Dawn dish soap and whatever. And you're like, hey, it's great.
Or how about
That's
leeches and
not,
cockroaches
I mean,
and
this year,
cicada shells?
everybody, if you really want to do this yourself in North America, there's 2 cicada populations that are going to be emerging this year. It's going to be massive and amazing. So find yourself some exoskeletons, everyone. But yeah, I mean, look at the components individually. We know that there have been years of research into nutritional components like turmeric, curcumin, trying to figure out what are the anti-inflammatory aspects and how are these these herbs that different people use, how do they actually work and what do they do and are they good and what are the like resveratrol. How many bottles of resveratrol do you need to drink to actually have it have any effect on you? And I think that is the problem. You can't have us, and I think you're right. For JAMA to put this kind of a study ahead, and for the person you spoke to say that this seemed like too important a result not to publish, you're right. That's faulty, and that study was faulty, and there needs to be more control, and that's lacking. If Eastern medicine practitioners want the stuff they use to get integrated, and if Western medicine practitioners are excited about that, let's do the science right.
Yeah.
Right.
But my problem is if an authoritarian government is demanding research from its scientist community to do a result, and they keep finding that result, and that result keeps ending up in Western papers because there's money involved.
And somehow it's always significant.
Yeah, like, no, that's bias, and that's not okay. That's low standard, and that's, like, at this point, like, this is the biggest fan of science you can get. And I'm like, I don't trust any research out of China. I don't trust anything in JAMA. I have to read the study just word by word and make sure that it's there if it's even in Nature. Because they're the ones that gave me the microbiome study on centennials that was people who couldn't have a birth certificate until 1950 and couldn't prove an age over 72. Like, they're doing a bad job. And there's great research that's out there. But allowing all of this. So, every once in a while we talk, we need more people in research. Yeah, I think we need less people in research. I think there's too many papers. I think you need to not be a grad student and publish a research paper. You can just do the experiment and know that you've gotten a result. It doesn't need to be published. The world isn't waiting to hear about your undergrad study on whatever it is.
I think that so I'll argue back against that. I think graduate students do need to publish and be a part of publishing, because by doing that, they learn how papers are written and they learn how the process works, so they can move into future stages of research and lead that research and lead the writing and know what they're doing. I think the issue is that the system is set up in such a way that we have hiring practices that are based on the number of papers and what journals you've published in the first place. How high impact are you to begin with? Oh, you published in Science, you get a job. What? And then from there, so people are, okay, so like Harvard, MIT, the big schools here in the US, like, it is super competitive and ridiculously cutthroat because everybody is tearing each other down to get the first result, to get the paper in Science or Nature. Like, that's like, because if they do that, then they get a job and that's the beginning of their career. Then once they get in as an early stage scientist, they have to keep publishing, publish or perish. If you don't put out high impact research, then you're not going to get tenure. You're not going to get grants. And if you don't get that, you don't have a lab. You can't support a lab. You can't support research assistants. You cannot do anything. And suddenly,
I think it's a good system because it keeps the pressure on, right? Of doing high impact research. But But you can't let them cheat getting there. You have to
That's
have a
the
gatekeeper.
no, that's the problem. The publisher parish forces people into situations where they make the wrong choice. They lie. They publish false data. They create things that will get them published so that they will get the job because maybe they come from a background that expects it of them. Maybe they have type whatever, type A psychology or whatever. They have to do this because they have learned in their life that the only thing to do is succeed, and success is publishing and getting a job and getting tenure, and that's what they do. And I think this is what's wrong, because what it creates is a friendly competition is great because it spurs people on.
Yeah.
But what is happening is we have created a system where it's so competitive and cutthroat, and it just people have to do it, and they'll do anything if that's what they believe is where they need to be, and you end up with people. I knew a postdoc whose paper had been rejected because one of her competitors in the same field was a peer reviewer, and that peer reviewer negatively commented on her paper, and then just right after published basically the same research.
Wow.
Yeah. So
that's pretty sketch.
It's sketch all the way to the top, and the reason is the universities and the financial motivation and how it all works. And if you get into a university, you then have to publish and you have to get grants. So you have to publish at a high impact so you have great ideas and you get the grants, and then you can hire people. And the more money you have, the more money you get to keep. But the university takes 60% overhead for many grants you bring in. So eventually you take the pay for peer review from the paper mill because you need some money, or you figure out how to do some crowdfunding. You figure out how you can actually make some extra money so you can pay your lab manager because you don't have any extra money because you haven't gotten a grant. And you can't do the research and you can't write the paper unless you have somebody else to help you run the lab. The whole system is a mess. Yeah, it's not the papers necessarily. That's part of it. The publishing part of it is just part of the system. But
yeah. It
anyway,
is, it's
thank goodness for YouTube.
A good gatekeeper, I think, at least when there weren't a lot of publications. And so to get published anywhere was a high impact because they didn't just take anybody to publish. They had real peer reviewers because they weren't throwing 500 papers that they wanted you to read. They gave you 3 a year.
Yeah. So let's go even further on this, though, and go back not just to the papers, but to the grants and to the paper and research that we've actually seen that has shown that there's bias in the reviewers for grants and papers if they know who is writing them. And people of color and women are not reviewed well in those situations, and they are less likely to get the grants. And so you're talking about gatekeepers, and you're talking about it historically as being a good thing. But in fact, I think what we are in the middle of seeing, and what you're witnessing, and what we're all witnessing with the breakdown of the publishing industry and what's happening with like these mills and everything is that there's a revolution taking place. For over 15 years, 20 years, people have been trying to fix publishing. There are a lot of good people who are trying to make it better. There are people who are taking advantage of a broken system, and because the system has been broken for so long. There is a revolution happening within the institutions, trying to turn over the diversity in these institutions, trying to not make it the thing where, the Black person is the person, they not only have to run their lab and write grants and do all this stuff. Oh, they also have to sit on the diversity committee, and they also have to sit on this other committee because nobody else has that experience. And so they're asked to do extra. And the whole system, like historically, we can't look back on it as the good old days.
I'm going to have to rename this segment.
Sorry.
This is now called, the new name for this segment, this is a new segment, is Justin Rants About a Problem, followed by less hope-filled words from Kiki.
No, and that's where I'm going to turn it around. And that's what I'm saying is that I think the revolution is needed and it's good. And I'm glad that people are starting to try to change things. They're trying to create better hiring practices. They're trying to create better peer review practices. The preprint archives are there for a reason, and people are trying to use them well. There are good open access journals and that are not paper mills. And there are people like Elizabeth Bick, who we interviewed, who was checking papers for false images and made up data. And there are people who are out there who are working to make science better. So yeah, I agree with you. I agree with your rant. It's broken. But I think that there are a lot of people who believe in it and not necessarily want to, be like, oh, let's fix the institutions and keep the institutions the same. Let's build something better. And I think that's hope. I mean, I have that hope that is where we are going right now. And unfortunately, as you go to something better, you have to tear old stuff down and it looks messy.
It does. And it's going to be incredibly hard to change because if you do, like anybody, if you show them, hey, you've been doing something wrong for a long time, their self-preservation is going to be, but there's never been a problem, right? It's not my fault. Oh gosh. Because you're also showing that a lot of the way that publishing has worked in the past has been inviting fabrication, has been inviting the paper mills.
And maybe wasn't controlled enough.
AI is going to really revolutionize this. And I think Web of Science could go a lot further than it's gone so far. But granted, they've done this just last year, got rid of 82 publications. So maybe I shouldn't be too harsh because it sounds like they're on top of it. But you could do simple things like
82 publications.
Show me a researcher that has been on more than 6 papers in a year. I can tell you one of the ones I was complaining about that I was trying to look at the peer review. It's a group of researchers that sort of end up on each other's papers all the time, sometimes in disparate things. One of them, I think, has been on 20 papers in a year. And I'm sorry, you have not contributed to 20 research papers.
Or you have. There is, about order, publication, author order, right? So right. First author is the person who did most of the work. Second author is usually the person who is like, their partner. And, they decided that, okay, first author did most of the writing or whatever. Second author maybe did this, a lot of the statistics or whatever. And it goes down the line. Last author is the person who runs the lab. And they didn't necessarily do any of the work, but they run the lab. And so their name is on the paper.
I'm not talking about the person running the lab.
No, but I just want to make it clear that there is a really interesting hierarchy, and it's very important. And it's the debate very often about authorship placement when a paper is being submitted is actually really important. Like graduate students, they really want that first authorship, at least on one paper. Because it shows that they did the work.
I was in charge of some things.
Yeah, but then you get the big labs who actually have a lot of people. Like there are labs out there that have 50, 60, like they have big labs working out there. And that primary investigator who runs the lab is on every paper that comes out. So he might, that he or she, I'm not gonna,
right.
they could be on 6, 10, 12
Totally.
papers a
And you have
year.
people that you're going to collaborate with. This is researchers at different universities across the planet or different labs who are tied through ODSI, which is
Yeah. So you're thinking about the ODSI thing. Yeah, going back to
I'm
that.
thinking about the ODSI thing, but I'm also I could be talking about a dentistry school in India where students regularly publish studies 2-3 a month and get them published in journals that are no longer accredited. But they advertise that their school has the most publications per student of any university for dentistry or whatever, which gives them a lot of prominence. And people were like, well, maybe I should go to that school where they're doing research on
it helps the PR
dental
and the
floss
marketing
with citations to themselves for a study on cotton candy's ability to withstand a high wind. Like a lot of times their citations have nothing to do with the study because it's a paper mill, so they're just grabbing things from other places where they're getting paid. But that's an example.
Yeah,
like run through that. Where did it come from?
Yeah.
Run through. Are they trying? Like, if you can come up with the algorithm for, is this a state policy of some level of internal self-propaganda that the Chinese authoritarian state has been pushing for? Is this a paper that confirms that, whatever view it is? Like, look at language, look at authorship, look at connections to pharmaceuticals. Like, we can uncover a lot of this. And guess what? You've published. The problem I have is there isn't any mechanism of consequence. There isn't any mechanism of like, hey, you know what? We can tell that every study you have worked on is in the favor of this.
Yeah, so I guess when you come down to like the government, like you're talking about the government pushed
or industry,
or industry. Yeah, I mean, but at the university level, we have seen a lot of stories recently where researchers have had their data like shown to be false going back. Like, there's been investigations into their entire history of publications, and like big falsifications have been pulled out, and they get fired. So
yeah, it can happen. It can.
And then you don't get grants anymore. Your grants are taken away, and then suddenly the NSF is like, "Well, you owe us money because you didn't do it right," and then you have to pay it back. So here in the U.S., I think at the institutional level for academics, when it is proven and the institution is like, "Yeah, we'll follow it up and we'll go with this because it looks good for us to actually do something."
Let me put it this way then. I think that if you are a medical student who has paid for authorship on a paper you did not contribute to, you should not be allowed to have a medical license. Period. Yeah. Flat. Like, you need consequences for this stuff. They used to have that test, that horrible test. People were like, oh, it's the big medical test. Everybody's afraid of it. Well, they got rid of it. It's now a pass-fail anyway. It's no longer has a score attached to it, right? And so now the talk is that you have to have these publications because that's the only way to differentiate yourself. You used to be like, I nailed it. I got like the highest score. Hire me because I'm smart and everybody. You haven't published. Yeah, I was too busy studying and learning my craft. Oh, that's a good point. Boom. Now there's nothing. There's a pass-fail. So now they have to do these public. But if somebody, and this is apparently where this those pay to play in the United States academic circle is the most egregious, is in the medical student.
Yeah, that category. In itself is a very competitive industry. Like,
yeah, but you want to be a doctor that helps people, and you're going to start your career showing unethical behavior.
Yeah, great.
Bye. There we go. So, this is the way. This is the way you separate.
So I think
those who really shouldn't be in the field. And I think AI can help.
I think listening to your rant and listening to your commentary, I don't think that we need to highlight any one country, any one kind of people, any one government, or, you know, any one industry. But I think the big take home from what you're talking about is that there are not enough controls. And right now, we are the publishing industry, because it needs money and researchers need money. There is a big hole, and it is causing um, in unethical behavior. It's causing false data to be published. It is allowing people to get places that they shouldn't, and it's going to hurt people, because we're going to end up with stuff like what was it, 20 years of Alzheimer's research that was based on a particular protein that, like, just recently they
Doesn't
discovered
exist.
that, like, the original data was not even solid. And so that is the take home, is that it's not just JAMA, that's one, but all of them, the Elsevier, the fact that there are paper mills, the fact that you can pay to be on a paper, like, all of it doesn't matter. Like, whatever country a scientist is publishing from doesn't, but if they're doing good
No,
science,
these are all
and
publications, and the ones I'm pointing out are all publications
I
in
know,
the United States.
yeah,
The publications are all in the United States.
but if it's good science, there should be somebody who can tell it's good science versus it's not good science, and that person is not going to be swayed by money or by influence. And I think that's the problem that we have right now. And yeah, that's what I was saying. I hope that's what people are trying to fix, and that's what we need to be fixed. And I totally agree with you. And
I do think it's fair to point out if egregious research is coming from certain sources, I think it's perfectly acceptable to point it out. I have no problem with that.
Right, but if you find a trend, you find a trend. But, like, that's just one trend, right? There are, like, probably others all over the place that you just haven't seen. So I think, yeah. Anyway, I agree. Science, we've got an issue because people need to make money, and that's where it's at right now. And we have, for years together on the show, we've talked about the fact that the pharmaceutical industry has hidden evidence, hasn't published results that didn't look good, and just published what they want to. We don't have, there's like one journal for negative results, but nobody publishes in it, right? What are we learning from each other? Like, when it comes to science, we need to learn from our failures, but instead people are making up wins, and that's screwing everybody.
Yeah.
We can do better.
We can, but hey, journals, you can reject more.
Yeah.
It's okay.
Oh man. Could you imagine if they did reject more, and just the editors like, no. And
but I'm trying to give you $4,000. No, thank you.
Then if you reject more, then you don't need as many peer reviewers. Yeah, I like this.
So it's bundled stuff too, because now it's like there's intermediaries that are like, hey, I have 20 studies to submit. Should I go to your journal or should I go somewhere else? And then the dollar amount is like multiplied for the, like, this is just, anyway, don't trust anything you read. Don't trust anyone you talk to. Don't trust anything you know. Don't trust any, I'm just gonna.
I'm going to caveat that with consensus and triangulation. So you can read one thing, and you can also read a bunch of things, but if you're only in like one little bubble of the world, you got to step out of your bubble. You have to go listen to other people. You have to read other things. You have to try other, like, One paper, we've always said this, the results from one paper are not gonna change the world, right? It is the results that get put together over time that change stuff, right? So yeah, no cherry picking, except if it's in the spring and there are lots of actual cherry trees and it didn't freeze and rain and it's nice cherries. And go ahead and pick the cherries and they're delicious. It's great.
I'm just constantly surprised though now of how much of just how bad this problem is. It's just like people like don't have standards.
It's worse.
People who you assume would have standards aren't having standards.
And the thing that to me, I think
everything for everything.
And then beyond that, it's not just the publications, then it's media and it's the blogs and the media sites that people go to for information that copy headlines and take, just whatever it was and they don't question it.
Right. And I already had lost confidence in them long ago. That's why they're not even part of the conversation. Like, they have no credibility on science whatsoever. Who cares what they're saying or talking about anymore? I don't, because they're trash. However,
but I've always
still thought, and I know that a lot of those side predatory publications that, XYZ journal that you've never heard of before because it's specific to this paper that somebody wrote, apparently. I get that those are trash. But now when it's in the mainstream journals on a regular basis, that's when you say it's gone. It's become critical mass, I guess is the thing, or something like that. Where now it's gonna be a runaway, the chain reaction of like you can kill science. You can. It's a living thing of people sharing knowledge and replicating knowledge. And if you continue to infect it with garbage that you want to publish and make money off of, you're going to kill the thing that feeds you, the thing that keeps us warm and healthy and keeps us from dying from the next pandemic. You're going to kill it because you're going to ruin the credibility, which science, if nothing else, is a process based on that credibility, the ethic of how you work, the unbiased truth. And it cannot stand for what you are doing. It should reject it. And it's sad to hear how tight of a bind researchers are in and getting their research and jumping through hoops. But on the other hand, there's a lot of people calling themselves researchers who aren't.
Yeah, agreed.
So, anyway, rant out.
And still more words of hope,
Oh yeah, we never got to the part.
I did give words of hope a little bit, but
some people, good people, are trying to do good things somewhere. It's just hard. Okay.
And in addition to your rant, I was reminded this last week when I lived in the 1890s here in Portland of how much science is useful and how understanding how basic concepts work can be used to your advantage to make your life better. We had things going wrong electrically. We had things going wrong with plumbing and other stuff, but because...
How come your plumbing, what you had on top of the
pipes,
power being out?
Pipes, don't let them freeze. It was... Seriously, it was
cold.
It was 29 degrees in my house. It was so cold. No heat. I had to trying not to let any pipes freeze and keep the water going. But it's because of understanding basic concepts of physics, electricity, understanding how current works. We were able to hotwire our heater to our heating system, our furnace to our generator. we now also have like these really cool new lithium battery backup packs that can put out almost as much energy as a gas guzzling generator, and they're quiet, which is really nice.
Utterly silent. Yeah.
And these things, it's engineering that put them together. Our house is put together through engineering, all of it. Engineering failed when the power lines fell down because of the trees.
You still haven't figured out how to engineer trees properly. That's it.
But it's science that helps us understand how to maybe keep the trees from falling down next time. It's science that has gotten us to the point where we understand how the current runs through, can run through lines and power a house. It's science that has allowed this house to stay slightly above freezing, so the pipes didn't freeze. It's science that gives us the batteries. It's science that formed the basis for the innovation that has allowed our society to move forward from 1890 to 2024. And so we need to foster that basic inquiry. We need to foster an environment that allows people to figure out how things work. And, and I agree. Things are weird right now, and they're breaking, but they're breaking all over the place. But I feel like we have a lot of good people.
I think science just needs to spend more time trying to kill vampires.
Oh, yeah. Okay. Werewolves? Just vampires.
No, I was thinking paper mills and pulp publishing. But those vampires that are feeding off of science or the people's desire to know more. As vultures, as vampires, drinking the blood of science for their own profit. Science needs to, as a community, figure out how to take them down.
Mm.
I think AI will help.
How to do better. Yeah, I think it will help. I think right now it's questionable and it's not, but yeah, it'll help.
We need a scientific blacklist, don't we? We need this person did, because who knows what nonsense. I want to know everyone who paid for. I want a list of everyone who paid for that have their name on a research paper, who has a medical license. I want every one of those names.
You can probably do that. Find databases.
And I want them open source publish. That is what I want to see.
AI can sort through all that data super fast.
What was it? The head of...
We've sorted.
Okay.
Yeah, we've sorted. We've ranted. We've had a whole thing.
I could do it again. That's what's scary. I was ready to go again.
How can I?
More ranting. Still not satisfied with my level of disgust.
Oh my goodness. Okay, don't. It's all right to be realistic, but have a little optimism, because there are good people in the world, and surround yourself with the helpers. Surround yourself with the people who make you laugh. Surround yourself with the people who challenge you and help you to be better. Surround yourself with people who help you find your joy. Let's do that. And on that note, I want to say thank you all for being here and helping me find my joy tonight. I needed that. And Justin, thank you. Everyone in the chat room, thank you for all your comments. And the Discord, of course, thank you for being there. Closing in on a tight 150. Thanks, Identity. Gord, Lore, thank you for helping to make sure that our chat rooms are nice, safe, happy places to be. Fada, thank you for all your help with the show notes and keeping us on track and reminding me to tell you whether or not we actually did have a show going on tonight. Keeping me going. Thank you. Identity 4, thank you for recording the show. And Rachel, thank you so much for your editing of the show. Just all of you. Can't do it without you. And especially, I have to say thank you to our Patreon sponsors. So thank you to Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, Rick Loveman, George Corris, Pierre Velazar, John Noam, Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan Shubrew, Sarah Forfar, Don Mundus Pig, Stephen Albaran, Cyril Myshak, Daryl Myshak. I have had wine this evening, sorry. Andrew Swanson, Fredus 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minish, Johnny Gridley, Remy Day, G. Burton, Lattimore, Flying Out, Christopher Dreyer, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, Lon Makes, EO, Adam Mishkon, Kevin Perishan, Aaron Luthenveld Calder, Marjorie, Bob, Paul D. Disney, not Bob. Hi, Bob. David Simmerly, Patrick Pecoraro, Tony Steele. I did it. I said all the names. Barely. Thank you all for supporting TWIS. We really can't do it without you. And if any of you out there would like to help support TWIS and keep us going, head over to twis.org and click on the Patreon link. We'd love your support. On next week's show,
we will be back Wednesday, 8 p.m. Pacific Time, Thursday, 5 a.m. Central European Time, broadcasting live from our YouTube and Facebook channels and from twis.org/live.
Oh, and I just forgot Blair wasn't here.
Want to listen to us as a podcast? Just search for This Week in Science wherever the podcasts are found. If you enjoyed the show, get your friends to subscribe too for more information on anything you've heard here today. Show notes and links to stories will be available on our website, www.twis.org. And you can even sign up for a newsletter that we promise we won't send.
And you can contact us directly. You can email me, Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, Blair at blairbaz@twist.org. Just put TWIS in the subject line so your email doesn't get spam filtered into one of Justin's pessimistic rants that, I don't know, you don't get ranted.
You can also hit us up on the internet.
Yeah.
Some places we are at TWIS Science, at Dr. Kiki, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes through in the night, please let us know.
And we'll be back here again next week. We hope you'll join us once again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.